Welcome to this lecture on the molecular evidences of evolution. Today, we are going to explore how the very building blocks of life, our DNA and proteins, act as a historical archive. By the end of this session, you will understand how scientists read these genetic sequences to uncover the deep connections between all living creatures, proving that evolution is a continuous, measurable process. Let us start by imagining you find two ancient books in different languages, but they share the exact same typos on the exact same pages. You would immediately know they were copied from the same source. In evolutionary biology, these books are the DNA sequences of living organisms, and the typos are random mutations that accumulate over millions of years. Because the genetic code is nearly universal, a specific DNA sequence codes for the same amino acid in a human, a bacterium, or a banana. By comparing these genetic typos, we can trace the history of life. Now that we understand what molecular evidence is, why is it so crucial? Sometimes looking at the physical shape of an animal can be highly misleading. This happens because of convergent evolution, where completely unrelated animals develop similar physical features simply because they live in similar environments and face similar survival challenges. Molecular evidence cuts right through this confusion. By looking directly at the genetic blueprint, which rarely lies, we gain an independent and objective way to test relationships. This is especially vital for understanding organisms that leave no fossils, like bacteria or soft-bodied creatures. Building on the idea of genetic blueprints, let us look at certain parts of the code that almost never change. Think of a car engine. You can change the paint color easily, but if you change the shape of the pistons, the car stops working. In biology, molecules like cytochrome C, which is essential for cellular respiration, and ribosomal RNA, which builds proteins, are so critical that any mutation is usually fatal. Because these highly conserved molecules change incredibly slowly, scientists use them to compare species that diverged billions of years ago, proving all life shares a common origin. While some molecules rarely change, others accumulate mutations constantly. This brings us to a groundbreaking idea proposed by Motuo Kimura in the 1960s. Imagine a massive lottery where most tickets are blank. Kimura proposed that at the molecular level, the vast majority of evolutionary changes are like those blank tickets. They are completely neutral, meaning they do not help or harm the organism's ability to survive. Because natural selection only acts on traits that affect survival, these neutral mutations are invisible to it. Instead, they accumulate purely by random chance over long periods of time. To understand how these neutral mutations accumulate, we can look at a simple mathematical relationship. Kimura showed that the rate at which a new neutral mutation becomes permanently fixed in a population is remarkably predictable. This leads to the elegant equation, K equals mu. Here, K represents the substitution rate, which is the number of new neutral mutations that become fixed in the population per generation. The symbol mu represents the neutral mutation rate, or the probability that a mutation occurs in a single individual. This formula shows that the substitution rate depends entirely on the mutation rate, completely independent of population size. Let us see what this formula means in a practical sense. Consider a population that experiences a specific neutral mutation rate, mu. Because these mutations are neutral, the size of the population does not influence how they become fixed. Therefore, if the mutation rate mu increases due to environmental factors, the substitution rate k will increase by the exact same amount. They are directly proportional. This steady, predictable accumulation of neutral mutations is profoundly important because it forms the absolute foundation for our next major concept, which allows us to measure evolutionary time. This predictable accumulation of mutations leads us to the molecular clock. If you know a faucet leaks exactly 1 drop of water every minute, you can figure out how long it has been leaking just by counting the drops in the bucket. The molecular clock works on the exact same principle. Because the neutral theory tells us that neutral mutations accumulate at a constant rate, we can count the number of genetic differences between 2 living species. This acts like a built-in stopwatch inside the DNA, allowing us to calculate exactly how long ago they shared a common ancestor, even without fossils.
We can express this stopwatch concept using a straightforward mathematical formula. D equals 2 times R times T. This linear relationship assumes mutations accumulate at a constant rate. In this equation, D represents the genetic distance, which is the total number of molecular differences we observe today. The symbol R represents the rate of molecular evolution, meaning the number of mutations that accumulate per lineage per million years. Finally, T represents the unknown time, in millions of years, since the 2 species diverged. If the time T increases, the genetic distance D will increase proportionally.
Before we move on, let us see this with a concrete example. Suppose we compare a blood protein between species A and species B, finding exactly 30 amino acid differences. We know this protein mutates at a rate of 1.5 mutations per million years. We want to find the divergence time, T. We substitute 30 for D and 1.5 for R. Multiplying 2 by 1.5 gives a combined rate of 3.0. Dividing 30 by 3 gives us 10. This means the species diverged exactly 10 million years ago, perfectly reflecting geological time.
Now that we have used this formula, let us understand where the number 2 comes from. What we are establishing here is why the total genetic distance requires multiplying by 2. When 2 species diverge from a common ancestor at time t equals 0, they begin evolving independently. Each separate lineage accumulates mutations at the rate r. So the first species accumulates r times t mutations, and the second species also accumulates r times t mutations. Because we are comparing the 2 modern species to each other, we must add both independent branches together. This simple addition gives us our final result, D equals 2RT. This diagram visually reinforces what we just proved. You're looking at a V-shaped phylogenetic tree representing the molecular clock. At the very bottom is the common ancestor, where time t equals 0. As we move upwards toward the present day, the lineage splits into lineage A on the left and lineage B on the right. Notice the small stars along both branches. These represent the neutral mutations accumulating independently in each species over time. The vertical axis shows the time since divergence, while the wide bracket at the top represents the total genetic distance, D. Because mutations happen on both sides of the V, we must account for both branches, which is exactly why our formula is D equals 2RT. Beyond tracking time, molecular evidence also shows us how entirely new traits are born. Imagine you have a vital recipe for baking bread and only one copy. You would never experiment with it because ruining it means starvation. But if you make a photocopy, you can keep the original safe while experimenting wildly with the copy to invent pizza. In evolutionary biology, this is called gene duplication. During cell division, an entire gene is accidentally copied twice. The original gene keeps doing its essential job, while the new duplicate is free to accumulate mutations and evolve entirely new, complex functions. This brings us to a stunning real-world example of gene duplication, the globin gene family, which carries oxygen in our bodies. Hundreds of millions of years ago, our distant ancestors had only one single globin gene. Through a series of duplication events, this gene was copied multiple times. Over millions of years, these copies mutated and specialized. Some became alpha globin and beta globin, which work together in our red blood cells. Another copy evolved into myoglobin, which stores oxygen specifically in muscle tissue. By reading these molecular sequences, we can trace complex modern traits back to simple beginnings. We have covered a tremendous amount of ground today. Let us reinforce the three most important ideas. First, molecular evidence provides an objective, quantifiable record of history written directly in our DNA, cutting through the confusion of physical anatomy. Second, through Kimura's neutral theory and the molecular clock, we can measure the steady ticking of genetic mutations to date evolutionary events with incredible precision. Finally, by studying conserved molecules and gene duplications, like the globins, we can trace the unbroken thread of ancestry connecting all life. You now understand how evolution is a continuous, measurable process inside our very cells.